The complete process takes several months, much of which is the data analysis, and we are still, to be honest, trying to work through all the data we have on these first 4 families, even though in some instances we've had that data for a while. Remember what I said about there are no perfect genetic specimens? That we all have 80 to 100 glitches in our DNA? Well, that means somebody with Moebius syndrome maybe has one glitch that's associated with Moebius syndrome, and then another 90 that have nothing to do with it. And so as you're sifting through the information, trying to figure out what's there, you've got a big challenge to determine what's causative and what's just along for the ride. So our hope had been that we would see recurring themes. If you found the same gene with a glitch in it, in 2 different families or 3 different families, then you'd really start to believe, well, that's probably connected. So far, we have not seen that. Each family has their own collection of glitches. And so as we look at those, we make up stories about, well, that gene looks like it would be a likely one to be involved. But we know so little about most of the 20,000 genes in the genome that you can make up that story about almost any gene if you really want to. So it makes it difficult at the present time to sift through the data. But we're starting down this path, and I think we're all pretty confident that with more and more information, it will start to fall together. I thought it would be useful, just as a way of showing you a little bit about what the data looks like, to tell you what we've seen in a couple of families, just as examples. So here's a family who came to NIH for the analysis, where 2 brothers and a sister are affected, and their parents are not. So we went through inheritance. I'm sure you're already saying, that's likely to be recessive, although it would be a bit unusual to have 3 out of 3 affected. It will happen. So this would predict, if it's recessive, that the parents would have a misspelling in 1 copy of the responsible gene. They'd be carriers, but they'd be unaffected. Whereas the kids who are affected would have that misspelling in both copies. And we have 3 kids to look at. So I'll show you that in this family we do have examples where that holds true, but I don't want to convince you in any way that we think that these are the right answers because of the problem that I mentioned of all the glitches that we're all walking around with and not being able to tell what's what. So here is what it looks like to the analysis that someone like Lori would be doing. Let me start here with the parents. Here's the DNA sequence, GCCAAG, and the top line is one of their chromosomes, and the bottom line is the other one. And here's the other parent over here. Now remember I said that information in DNA has to somehow be translated into protein? Well, it turns out it's 3 letters in the DNA and also the RNA that gives rise to a single amino acid. So GCC means alanine, and AAG means lysine, and GAT means aspartic acid, and so on. Now in this instance, both parents have 1 chromosome, marked in red here, that have 6 extra letters in the DNA. That CCAAGG doesn't appear in other people. We haven't found other people who have that. It's because it's 6 and not 5 and not 7, what it does is to insert 2 amino acids, an alanine and a lysine get stuck in the middle of this protein product, but otherwise the protein would be entirely normal. Now again, both parents happen to have that insertion of those 6 letters, and it turns out in this family all 3 children have inherited the yellow copy with those extra 6 letters, so both copies of this particular gene have that in the affected kids. So that would be a pretty good story for the cause of a recessive disorder, but the problem is we have others like this. So I can't tell you that I'm particularly confident that this is the explanation of Moebius syndrome in this family. I'm not confident at all, but it's the kind of information that we're looking for. Now, if that same gene turned out to have either this glitch or a different glitch, slightly upstream or downstream, but in a different family, then I'd start to get more convinced that we were onto something. Right now, it's too early. I'll show you one other family. This is often the case for Moebius syndrome, where you have unaffected parents, and out of nowhere, the child ends up with the condition. So what's going on there? We would call that a sporadic case. maybe that's not the best chosen word, but physicians have many words that aren't very well chosen. So it's a sporadic case. It could be recessive, of course. The parents might be carriers. And if they've only had one child, well, you wouldn't necessarily know that it's recessive unless they had lots of kids and you started to see it pop up over and again. Or the other possibility, and we have to think about this, is could this be a new misspelling? When DNA gets passed from parent to child, each of us ends up with about 60 spelling differences that our parents did not have. Because DNA is hard to copy. When you've got 3 billion of those letters, occasionally something doesn't come out quite right. And that's probably important, or we would still be bacteria somewhere in the slime. So this is part of the way in which evolution does its job, is having a slight chance of some changes. And so knowing that we all have these new mutations, these new changes, new spellings in the DNA, you might be particularly interested if you found in somebody with a genetic condition with no family history that they had one of those in a vulnerable place. If that were to be the case, you would predict the parent's genes would have the normal sequence, but the affected child would have a difference. Again, because each of us probably has 60 or 100 of these, it's pretty hard to get excited when you see one, but I'll show you one anyway. So here is a family where both parents have the expected sequence in this particular part of this particular gene. This is an exon 14 of all things. You can see the normal sequence. TTC codes for phenylalanine, CGG for arginine, AAG for lysine. Mom and dad both have both copies, just as you'd expect. Oops. But when you look at the affected son, there's a single letter change in one copy. So instead of a C in that position, there's a T. And that actually changes the coding of that, because CGG codes for arginine, but TGG codes for tryptophan. So that means the protein that's made here is going to have a slightly different function. Could that be the cause of Moebius syndrome in this young man? I don't know. It's certainly on the list of possibilities. But again, because there's a background here of these events in all of us, I'd be very reluctant to point to this and say, aha, we have the answer. Bottom line is, if we're going to get those answers, I think it's going to be really critical to have more and more families willing to go through this process of examination and DNA analysis and see if we start to see recurring examples where the same gene pops up in more than one family. So what lies ahead for genetics research on this condition? Well, I think exome sequencing will and should be conducted on more families. We at NIH have the good fortune to have very effective abilities here in a fine sequencing center, and these folks who are capable of analyzing the data, and people like Lori who are very dedicated to this. So we hope to be part of that in collaboration with other groups, particularly the Boston Children's Group and the Mount Sinai Group and others. Such as Toronto, anybody who's got a willingness to be part of this team, and there are lots of us, I think, would recognize that there's much to be gained by working together. The goals will be to try to understand if there are genetic contributors to Moebius syndrome in an individual. What exactly are they? Can that be utilized to provide more accurate diagnosis and a prediction of the likely course? And maybe some new ideas about therapeutics, or at least the ability to identify what's likely to work for which person in a more effective, accurate way. But again, I've been talking about genes. I want to emphasize again that I have no confidence that genes are going to be the whole story. I suspect they won't be. And we, at the same time that this kind of work is going on, need to think about ways in which we could identify other things that contribute, such as environmental exposures, which have not yet been identified, but perhaps with more work could be. I think sometimes people tend to assume that researchers are either in the genetics category or the environment category, and in fact, in this circumstance, we clearly need to think about both. Most importantly, just to wind this up, I think we have a great opportunity, and this foundation has been a wonderful supporter of this idea of working together, of working as individuals who are affected with the condition, their families, researchers, lots of other individuals who have gotten interested in this condition for all of its complexities. We really have a team here in Philadelphia, And that is great, because if you really want to see something happen, it's one thing to have a single person stand up and sing some beautiful music. It's even more impressive if that is part of a group that sings together with a full orchestra. And my hope is that the kind of thing that's going on here in Philadelphia will add further to what has already been happening in a wonderful way because of the Moebius Syndrome Foundation, to bring together all of the people who have an interest in this condition and allow them to form the kind of team that's really going to make a difference, ultimately getting you the answers that you are hoping will emerge. Because I'm pretty excited that in the next short few years of time, there's going to be a lot more understanding of what causes this condition and what we might do about it. So I thank you for your attention. It's been wonderful to be here with you. I'll be glad to answer a question or two if people would like to pose them. I'm not quite sure if that's what the organizers had in mind, but thank you very much.
Well, thank you for that. I have a question about links between the genetics.
Oh, we do have microphones in the audience. My goodness. Thank you, Don.
And I'm curious about
I'm not sure it's on, though. Can we get the mic?
Any association with what?
There it is. Hold it very close.
I'm interested in any associations or understanding between the links between Moebius and autism, particularly early development.
So I think there's a strong interest in trying to understand what has been reported as a connection between Moebius and autism. I've read a couple of those papers, but I'm far from the expert. I know there are issues there that people are debating about the degree to which this is autism in the form that you would see in other circumstances, or whether some of it is in fact on the basis of the effects of Moebius syndrome on facial expression and how that impedes social interactions, which are a big part of the autistic spectrum. So I'm not the expert, but I think that is a topic of great interest for people who are trying to see what could be learned on an even larger stage. There's a general sense that when you study a rare disease, you often bump into things that have implications for much more common conditions. And if by studying Moebius syndrome, we also got an insight into autism, which is becoming a truly frighteningly frequent condition in the US. Now, 1 in 88 individuals diagnosed with autism spectrum disorder. That would be a wonderful dividend for all the research that's going on. There's somebody over here who wants to. Question?
Good afternoon. I wanted to know how would I go about participating in the research center?
So at the moment, I think your best bet is to interact with the groups who are here from Dr. Engel's lab and Dr. Jabb's lab, who have set up opportunities to interact with people at the meeting. At the moment, we're primarily being led by their suggestions. If there are individuals that particularly would benefit from the very intensive analysis and evaluation we do at NIH. So I think your best doorway would be through those folks. David Hunter standing in the back. Maybe somebody's here from the Jabb's lab who could stand up and indicate your availability. I don't see them at the moment, but they're all over the place here in the next couple of days.
Phil Donahue retired.